Okay, good morning, epidemics. Hope everyone's doing well today. Have you guys found the lizard in the big picture?
Oh no.
Nope.
I think I have.
I think I see it too.
It's right in the center.
Yeah, it's right
Yeah,
here,
right there.
right there in the center. Yep.
I'm colorblind, so I still can't see that.
Oh no. So not sure if that'll help, but it's right in here.
I'll suspend disbelief for a second.
Okay, so announcements. I'm changing up the schedule starting this week. So instead of posting quizzes and journal entry prompts on a Monday, I'm going to start posting them on Friday. You'll still have a week to do them. So let's see. Yes, so your quiz and your journal prompt will be posted on the 10th and will be due on Friday, February 17th. All right, we've been talking about HIV. Does anybody have any questions or thoughts? Anything you want to talk about? All set. Okay, good. So worldwide at this point, we've been talking about discrimination. And I want to make sure that you guys understand that there's still LGBTQ plus discrimination and HIV positive status discrimination all around the world. Last time I checked, which I think was last year, there were 48 countries that ban entry to people who are HIV positive. There are 70 countries where it is illegal to be LGBTQ plus. In 11 of those, they have the death penalty still active for anyone who participates in homosexual acts. And 6 of those countries are actively killing people under this law. So you can still, Eric, yes. Go ahead, Eric.
I thought I was unmuted. How would you know if someone was HIV positive entering a country? Because that's not on your like documentation for like immigration status and stuff like that.
Right. I don't know about immigration. They may require testing before they would let you immigrate there. As far as traveling, I don't think that's really enforceable, especially in the modern days where we can get, with medication, people can get below a testable or a measurable threshold of virus. We don't have a cure yet, but we can get that viral load down so far that it doesn't show up on tests.
Would this be more like if they live there, they can't access any medication or be diagnosed, stuff like that?
Yes,
Yeah.
yes, you couldn't access medication. They probably, well, it varies dramatically. So there are countries where you cannot get access to medication. But the idea here is if you're traveling to another country and you need medication or medical services, you can be caught out and then sent home. So if you're traveling for vacation, you know, that's just traumatic and probably expensive. But if you're traveling for longer time periods, like you're going to do business in a country, and you may be doing business for a month. That could cost you a lot of money. That could cost you your business connections, and you'd probably get put on a list saying you're not allowed back in.
Makes sense.
Mhm. So a lot of people, both in the past when HIV was new and even today, don't understand how it's transmitted, and they are really scared of catching HIV. So people would be concerned, especially in the early days, that you could catch HIV by sharing a drink with somebody. One of my friends in college, his mother absolutely went ballistic when she found out that there was a member of their church who was HIV positive. And she didn't think he should be allowed in church because she thought, I don't know, he was evil or contagious, or she would catch it. And in fact, she switched churches, and she was very, very religious. So switching churches was a big deal. She switched churches because of this. One person with HIV in her church. In the early days of HIV was a death sentence. We didn't have treatments. And we had all kinds of problems. We had hospitals that would not treat HIV positive patients. Morgues would refuse to handle the bodies. Schools would refuse to admit children with HIV. And this was a time period, my parents went through it. I was a kid. So I've got some stories about this time period. A friend of my dad had a son who was HIV positive, and he was very sick, and they called EMTs, called an ambulance. Emergency medical technicians came out. The EMTs did not want to treat him because he was HIV positive, and they were afraid they were going to get HIV from him. They told this child's parents that they should just let him die. Can you imagine the horror of calling emergency medical services and having them say, no, you should just let the kid die? So dark, dark times. Okay, so when we talk about our diseases, our epidemics, we're going to see a lot of recurring themes. Overall, we're going to see that epidemics are spread by globalization and travel. The more people move around, the more likely an epidemic or pandemic is going to start. We see that people in poverty and people in oppressed cultures are more likely to get diseases and die of them. Wealthy people are less likely. A lot of that is access to healthcare. Lack of access to healthcare contributes to pandemics. And this is not just in the poor and oppressed. This is everybody. If you live in a country like the US, where there's not government-funded healthcare, a lot of people cannot afford to go to the doctor. When that is the case, the disease tends to run rampant in that country. It tends to kill more people in that country, even people who do have healthcare, because they're exposed to the disease more often. We see that with the COVID pandemic. The US is the one country in the modern Western world that doesn't have government-funded healthcare. We also have a much higher death rate for people who have COVID than the rest of the modern westernized countries. The fourth major theme is misinformation. When we talk about HIV, until very recently, advertisement for medication and information about HIV showed white men. They didn't show women. They didn't show African American men. They didn't show, I haven't seen any that show Hispanic men. It's not just white people. It's not just men. And it's not always transmitted by sex. So it's not always even adults. With HIV, one of the contributing factors around the world is homophobia, which is why I ranted on it so much, and cultures that are what I would call repressed, that aren't comfortable talking about sex. And this includes, the US has gotten much more comfortable talking about sex and less homophobic, but there are places in the world where they're still very homophobic, still very repressed, and people don't get the information they need to deal with prevention or treatment of HIV. Specifically with HIV, condoms reduce the risk of transmission. They work very well if they're used consistently. Most people don't use them consistently. So we say the risk of transmission is decreased by around 70%. But there's still stigma talking about using condoms. There are still people who won't discuss that, and there are some men who refuse to wear one. Even in this country, polite people do not talk about sex in public. I'm not polite. Educators, biology teachers are often nervous about talking about sex. And some doctors are nervous about talking about sex. So we get public messages that are confusing, and we don't get corrections from educators and doctors. In pop culture, we've got this message that sex is between men and women. And up until pretty recently, it was very much focused on husbands and wives, married couples. The early HIV education information talked about avoiding the exchange of bodily fluids. Now, I don't know about you, but that's vague. It should have been safe sex. But of course, we were much more repressed back in the 70s and 80s. I mean, what does it mean when you say don't exchange bodily fluids? No kissing? Don't cry on somebody's shoulder? Don't win the championship basketball game and then go hug your sweaty teammates? Yeah, so that was very confusing. It wasn't just gay men who were getting HIV, and it's still not. The other high-risk group that we discovered early in the AIDS pandemic was IV drug users. Well, guess what? This is another group we can shun. The idea was that if you got HIV from IV drug use, you got what you deserved. because we're very, there are a lot of people who are judgmental and vindictive. And while IV drugs are generally across the board pretty nasty drugs that you shouldn't do, a lot of people still do them. Lots of illegal drugs are illegal because they can be dangerous or harmful. So methamphetamine, heroin, those are really nasty, nasty drugs that can do a lot of damage to your psyche and your body. Other drugs are or were illegal for government moralizing, like pot. Marijuana is not very dangerous. We're seeing states that are making it legal because it's not really that dangerous. It's not as dangerous as cigarette smoking, for example. So IV drug users. Okay, we've got the gays, we've got the IV drug users. These are oppressed cultures. But in the early 1980s, we started seeing a third group, and this third group really started changing things up. And this group was hemophiliacs, people with hemophilia. So hemophilia is a blood clotting disorder. It can be life-threatening. And one of the more common types of hemophilia is treated with something called factor VIII. Factor VIII is something that people who don't have hemophilia naturally produce. It's hard to manufacture in the lab, so the best place to get it is from donated blood. So what they would do is they would pool blood samples, extract the factor VIII, and then inject that into people who had hemophilia. So you're getting this blood from multiple individuals every time you need to be treated. This is a very, very, very high risk for transmission. By 1984, half of all people with hemophilia were HIV positive. This dramatically increased public support. This is not a marginalized group. This is not somebody that we can judge and say they deserve it. This is people who are sick, and it includes a lot of children. Any blood transfusion with HIV positive blood has a very, very high risk of transmitting HIV. Even once we had tests, blood banks in the US did not start testing blood donations until the cost of lawsuits from people who got HIV from blood donations became higher than the cost of testing. It was all a money thing. And then in 1985, 1986, something happened that really, really started to turn the tide on the whole HIV pandemic and the American perception of it. And it's all about this kid. This was Ryan White. He was a hemophiliac. He was HIV positive, and in 1985, he was banned from public school. The first day of school, they were like, well, we are required to educate him so he can listen to classes over the phone. We didn't have the internet, so he was literally listening on a telephone. And he's fighting this. He wants to go to school. In November, the Indiana Department of Education says, no, you must admit him to school. You must allow him in school. The county school board votes to appeal the ruling, 7-0. The Indiana Department of Education again says White can attend schools, and we're going to have a county health officer to look at him and see if he is fit to attend school. And the health officer said, yes, he's healthy enough to come to school. It's not a high risk for the other students. And at this point, it's February. And the school board tries to fight it, and the county judge says, no, I'm not going to issue an injunction against this kid going to school. The state Department of Education has ruled that he can go to school. He can go to school. February 21st is the first day he was able to walk into the classroom during the 85-86 school year. By that afternoon, a different judge had granted a restraining order barring him from school. On March 2nd, people held an auction in the school gymnasium to raise money to continue the legal battle to keep Ryan White out of school. In April, the case goes to the circuit court. So the circuit court is a court that covers multiple states. It'll be an area of the country, so it's a higher level than the state courts. In April, his case was presented to the circuit court, and on April 10th, the circuit court judge says, no, we're getting rid of the restraining order. He's allowed back in school. And the Indiana Court of Appeals declined to hear further appeals. So, on the day in February when White returned to school for the first time that school year, 151 out of 360 students were sick that day and didn't come. Almost half of the students didn't show up. He was healthy enough to have a job as a paper boy. I know we don't have these anymore, but it used to be a thing. People on his route canceled their subscriptions. Although the school had to allow him to attend starting in 86, so for the 86-87 school year, he had a separate restroom just for him, and he was required to use disposable utensils in the cafeteria. He was not allowed to participate in gym. Threats of violence continued against Ryan White and his family. Eventually, his family was forced to move because people literally shot up the house they were living in. In 1990, he died of HIV at age 18. Having this public teenage boy be the face of HIV really started to change public opinion. And we started to see more funding for research, more support for that research, but there was still an awful lot of discrimination. Do you guys have any questions or thoughts about Ryan White and his, I mean, he must have been an amazing kid to go through all of that while sick. No.
Do you know if like the stigma against him was mostly by other students, or was it by like parents who were concerned for their own children, pulling their kids out of school or stuff like that?
It was largely pushed by parents. but kids learn from their parents, and there was definitely some discrimination against him from other students. But it was definitely more on the parents' side than on the kids. All right. So this is a confusing image. What we're going to talk now, we're going to shift from, well, not completely shift away from discrimination and bias, but shift more towards the biology for a bit. And I want to talk about how the virus was discovered. It takes something called shoe leather epidemiology to figure out how a disease is spreading and get an idea of how it is being spread. So what this means is that epidemiologists interview people who are infected and try and find out how the disease is spreading. Did you eat at any restaurants? Did you go to a public event? These sorts of things to try and get an idea where the different people interacted to spread the disease. And the interviews found overlapping circles of sexual partners, which suggested an STD. And it didn't respond to antibiotics, so it was probably a virus. This early work gave us the term patient zero. So patient zero is conceptually the first person to be infected by a new disease. It's not a great term because a lot of times when we have spillover events, we can have multiple spillover events in a common area. So it's not necessarily one person got the disease from one chimpanzee or one bat. It can be 3 or 4 people got it from 3 or 4 chimpanzees or 3 or 4 bats. But this was based on a study that looked at a cluster of 40 infected men and tracked down their sexual interactions. They were focused initially on San Francisco and Los Angeles, and then added in New York City shortly thereafter. And the earliest patient they could track it back to was designated patient O, not 0, O, the letter. And it stood for out of California. So it meant somebody who wasn't in California. It later got changed to 0, and we started looking at other individual cases. So this is this cluster of reproductive interactions among these guys. And the different colors, and I know somebody said they're colorblind, and so you can't see this, list the different infections that they showed up with. That's not all that important. Patient 0 had sex with patient 9 from Los Angeles, patient 8 from Los Angeles, patient 3 from Los Angeles, and patient 6 from Los Angeles, as well as patients 3, 4, 9, and 15 from New York City. Patient 3 had sex with patient 1 in Florida, patient 2 in Los Angeles, and you can see how it kind of spread out. You know, sometimes it's just spreading to one other person we knew about that we studied in this particular example. Sometimes, like Florida patient 1 definitely spread it to 4 other people. Patient 17 in New York spread it to 4 different people. And back and forth we go. Unfortunately, the identity of patient 0 got leaked. He turned out to be a flight attendant, hence the having sex with men in multiple cities. He traveled a lot and he died of AIDS in 1984 at age 32. He was blamed and vilified as the person who brought HIV to the US. Yeah, he wasn't. He did spread it around. But retrospectively, looking at blood samples, the HIV pandemic was firmly established in the US before the spread started from this patient. There's a book called "And the Band Played On" that called him out by name and definitely brought a lot of the idea that he was to blame for the epidemic. It's ridiculous to blame one person unless they're going around with needles and injecting people. So the race was on to figure out what is the virus. And it really came down to these two guys here. Oh, I hate pronouncing French words. I'm not good at it. Luc Montagnier, maybe, and Bob Gallo. Luc Montagnier was at the Curie Institute in Paris. Gallo was an American virologist. In 1983, Montagnier published that they had found a virus. They called it LAV. And he said, I think this is the virus that causes AIDS. Bob Gallo got a pre-publication copy of Montagnier's paper where he was going to publish this data. He was what we call a reviewer. So scientific publications require researchers to submit their paper. The paper is then passed on to several other experts in the field. And these experts read the paper and say, yes, it looks like the science was good. Yes, it looks like their conclusions make sense. Yes, you should publish it. Or, you know, I'm not sure this experiment was done correctly. I'd like to see it repeated. Or I don't think the results of their experiments justify their conclusions, this kind of thing. So Bob Gallo reviewed the paper. He was afraid that somebody else was going to get credit for finding this virus. And he argued with the CDC, the Centers for Disease Control in the US, to try and prevent the French lab from getting any blood or antibodies or other specimens from America. He claimed that the French sample that they had been working with was contaminated, and that what they had found was the contamination, not the actual virus. Bob Gallo was a bit of a jerk. In April of 1984, Bob Gallo published a paper saying, I have found the virus. It's HTLV3. Notice that neither of these names are what we call it today. In 1985, other researchers compared HTLV3 from Gallo and LAV from the French lab and said, hey, wait a minute, these are almost exactly identical to each other. And then people started looking into it and realized Bob Gallo had gotten samples from the French lab, from Montagnier's lab. They were both of these men were trying to get a patent for an antibody test to detect the disease in blood samples. And the legal dispute got so bad and was holding up progress that French President Jacques Chirac and US President Ronald Reagan had to forced the labs to share credit. And these two presidents renamed the virus HIV so that neither one of them could claim sole credit for discovering it. In 2008, a Nobel Prize was awarded for the discovery of this virus. It was only given to Montagnier. So the Nobel Committee said, Bob Gallo, yeah, no. Montagnier found it first. You got yours from him. Okay. By the time Jacques Chirac, the French president, and Ronald Reagan, the US president, forced the labs to share credit so we could start to develop an HIV test, 50,000 Americans had died of AIDS. So, woo, lots of words on this slide. We're focusing now on biology for a bit. So the virus can be spread. Let me put my text up here. By blood, semen, and vaginal secretions. Not by saliva, not by sweat, not by tears. And it is spread through unprotected sex, i.e., sex without a condom, blood transfer, transfusions and transplants, sharing needles, that's drug use, and what we call vertical transmission. Vertical transmission is from mother to unborn child or during childbirth. So let's look at our risk and get an idea of what's the most risky thing here. Receptive penile vaginal sex. So the person with the vagina, if the person with the penis has HIV, it gets transmitted about 8 times per 10,000 exposures. That's not too bad for one interaction, but people have a lot more sex than that. So it does spread a lot that way. Now, rape doubles the risk. It doubles the risk because rape tends to cause damage to the vaginal wall, and then the semen can get across that barrier into the bloodstream carrying the virus. Blood transfusion, 9,250 cases per 10,000 cases. That's more than 9 cases out of 10. So blood transfusion, receiving an infected blood transfusion is very, very, very high risk. Our blood supply nowadays, really well tested. We've got a good idea whether or not the blood has HIV or does not have it. Blood donation, 0 risk. You can donate blood. You're not going to get HIV from donating. And in the US, blood transfusions, like I said, they're very safe today. Sharing needles, 63 per 10,000 exposures, so quite a bit more than receptive penile vaginal sex. The rate is lower for accidental needle sticks in healthcare settings. So it can happen that usually a nurse is taking a blood sample from a patient and accidentally sticks themselves with the needle. We've got technology these days to help reduce that risk, but there is a risk of spread that way. And then there's receptive anal intercourse. And the receiver has a risk of 138 out of 10,000 exposures. So quite a bit more. That's actually more than IV drug users sharing needles, twice as much. And again, anal intercourse is more dangerous because you tend to damage the tissue lining the anus. And whenever you damage that tissue, you're giving an opportunity for the virus to get into the bloodstream. Condom use reduces the risk by 80%. That's very significant. PrEP, which is pre-exposure prophylaxis. Pre-exposure prophylaxis is something you would take if you know you're going to be having sex that's risky, or you know you're going to be having sex with someone who is positive. And that, along with condoms, brings the risk down even further, reduces the risk by 92%. If you've been exposed, post-exposure prophylaxis reduces risk by about 80%. And children, the vertical transmission, we can have less than 2% risk if there is treatment for the mother and for the child when it's born. Anything that causes open sores or wounds increases your risk. So herpes, for example, increases your risk because the virus has to get into the bloodstream. This is an image of the virus. So this particular virus has RNA for its genetic material. It's got a very complex shape. It has some enzymes inside the middle of the virus. It has some surface proteins, and it also has a lipid membrane, similar to the membrane of your cells. Okay, questions or thoughts on this information? I know it's a lot of numbers, but I wanted you to have an idea of the ways it's transmitted and which ones are riskiest, and how to reduce risk. Okay. So now we're going to get to some more complicated virology. The HIV virus, I mentioned, I'm going to close my chat window so I can write on the screen for a second. So the virus has a protein on the surface called GP120. These names suck. I gotcha. I know that. GP120 binds to a surface on certain immune cells, specifically a type of white blood cell called CD4. So we say the types of cells that can get infected by HIV are CD4 positive, meaning they have this particular protein on the cell surface. CD4 is an important protein for immune system cells to communicate with other cells. So they release molecules that bind to this CD4 protein to carry information from one cell in the immune system to another cell in the immune system. HIV then binds to what we call a chemokine co-receptor. Don't worry about that phrase, a different receptor. We're just going to call it a different receptor. That receptor can be CCR5 or CXCR4. Oh my God, more abbreviations. The only abbreviation I'm going to ask you about is CD4. The others I'm just, I'm going to talk a little bit about CCR5 and CXCR4 because there's a way that there can be some resistance to HIV infection related to those. But I'm not going to ask you for details. I'm not going to ask you to come up with the names yourselves. All right, so it binds to the second receptor. When it does that, the lipid membrane around the outside fuses with the cell membrane, and all of the proteins, enzymes, and genetic material get injected into the host cell. So now the viral RNA and the proteins are inside the cell. And it's using proteins on our immune cell surface that are required for our immune cell function to get into our immune cells. Now, the next thing the virus needs to do is trick the host cell into making copies of its genetic material and to make some of the proteins, like the proteins that make up the virus, and release new virus particles. So your cells work with DNA. We do have RNA, but that's not where we hold our genetic information. So the first thing the virus does is copy the RNA and make a double strand of DNA. So it converts that RNA genetic material into a DNA version. The DNA version goes into the cell nucleus and inserts itself into your chromosomes, your genetic material, your genes. What this means is that that cell is permanently infected because the virus now has HIV viral DNA in your DNA. And there is no technology available today that we can use to get it back out. So then your cell does normal cell stuff, and it looks at this HIV DNA and makes the virus proteins and makes the genetic material for the virus, the viral RNA, and those all get clustered together and assembled, and a new virus gets released. Okay, so these cells that are infected can continue to function for a little while. One of their functions is to make copies of themselves, to make more cells like them. When they do that, all the new cells are also infected with the virus, have the viral DNA. Each infected cell can produce billions of virus particles. Virus particles are called virions. It can produce billions of these every single day. And the process of requiring the host cell to produce this much virus eventually kills off these CD4 cells. And it kills them faster than they can be replaced. Okay. Because the virus has such a high number of new RNAs, new viral RNAs, and new virus particles produced every day. It's going so fast, there is what we call a high error rate or high mutation rate. This helps the virus develop drug resistance quickly. If you're producing from one infected cell billions of virus particles, if 1 million of those have a mutation that make them unable to infect cells, so what? You've got, over 900 million more viruses that can infect cells. So this drug resistance rising so quickly means that the best way to treat our HIV is a multi-drug cocktail system. And we have drugs that can block various steps of this process. We can block the process where the virus, once it's attached to the receptors, is actually able to inject its genetic material into the host cell. So that is a category called fusion inhibitors. Between my undergraduate work and my graduate work, I spent 5 years working on HIV, and we specifically were trying to inhibit the fusion process. And one of the drugs that we worked on is on the market. Then there's a step where we're copying the RNA from the virus to a DNA form, and that's a step that we can inhibit. We can inhibit binding to this secondary receptor on the surface. We can inhibit the assembly process of the new virus. So all of these steps we can inhibit. And when we, and integrating into your host DNA. When we make a drug cocktail, we try and include drugs that inhibit at multiple different steps to really help make it hard for the virus to successfully make new virus. Now, I said I was going to come back to CCR5 or CXCR4. I'm going to erase what I've written real quick. There are people who are missing CCR5 or CXCR4. These people are resistant to infection. If you don't have, especially the CCR5 one, if you don't have CCR5, it is much harder, possibly impossible, for the virus to infect your cells. We think missing CCR5 originally came about because it provides resistance to smallpox, another virus. But unfortunately, it makes people more vulnerable to other pathogens. If you're missing that CCR5, things like West Nile virus and various forms of tick-borne encephalitis. Encephalitis is inflammation of the brain. Not good stuff here. Whew, lots of information on this slide, sorry. Do you guys need me to repeat this, go over it again? Do you think you understand what happens during infection?
I understand.
Thank you. Anybody else want to chime in?
Yeah, I covered a lot of this in my biology class last semester.
Awesome. That's great. All right. This is a graph showing what happens when people are infected with HIV. So the x-axis here, it starts out in weeks and then changes to years. So the first 12 weeks and then up to year 11. The y-axis on the right side is how much viral RNA can be found in a blood sample. It's a way of measuring how much virus is in a person. On the left side is CD4 positive white blood cell count. So how many of those CD4 cells you have in your system. And so what we see that there is a primary infection phase. You've been exposed to HIV, it's gotten in your bloodstream, it's infecting your CD4 positive white cells. And this early phase, we call this an acute phase. Your CD4 white blood cell count drops dramatically. Your viral load rises dramatically. And you might have symptoms kind of like the flu. So head cold, fever, fatigue, that kind of stuff. But then you kind of start feeling normal again. And so the next period is called latency. And during latency, we don't have, so this is our line for virus concentration. It goes up slowly over the next 7 years. The CD4 cell count, which is this line, goes down, and it goes down basically for the rest of the course of your infection unless you get treatment. At this point right here, I'm going to draw a line to mark it. Right about the 7 to 8 year mark right here, your CD4 positive blood count falls below a certain threshold. And your immune system is no longer able to keep up with diseases. Your immune system is now impaired. And we base that on this CD4 count specifically. At this point, we start seeing opportunistic diseases. And this period can last 3, maybe 4 years. And at that point, the patient dies. So stage 3 here we call AIDS. A couple of important points. During this primary infection phase, HIV, especially very early in this, the first 2 weeks or so, HIV tests may or may not be accurate. They may not detect the virus. They'll detect it when it's at this very, very high peak, but that's week 4, 5, or 6 right here. A lot of people don't know they're infected at this time. If you don't know you're infected and you're at this phase where you've got a lot of virus, you are highly infectious and highly likely to transmit the virus. As our CD4 cells decline, the body loses its ability to suppress the infection. Now, this graph shows what happens in untreated patients. With treatment, this latent period can last for decades. We don't know how long it can last, but there are certainly people who have been in the latent phase of infection for decades. AIDS itself tends to be a dramatic decline over the last few years of a person's life. With modern treatment, like I said, people can live for decades. Early treatments for HIV were hard to deal with. They had lots of side effects, nasty side effects. And you often had to take multiple pills a day on a very tight schedule. So you would be on possibly 5, 10, maybe more pills, different pills every day. Some had to be taken with food. Some could not be taken with food. Many of them could not be taken at the same time of the day. So you had to have this schedule and you had to stick to it. It's also very, very expensive to get treatment. Modern treatment, we have multi-drug pills. And these multi-drug pills means that some people may only need to take one pill. And I've seen, although I haven't done the research on this, that we're developing other treatments that you may not even need to take a pill daily. Modern treatments are still expensive. The good news is people on these modern treatments can have a nearly normal life expectancy, which is great because the original life expectancy for somebody with HIV was usually 7 to 10 years after they found out they were positive. But there are still nasty side effects: liver damage, nerve damage, increased risk of heart disease and heart attacks. Some patients can get to no detectable HIV in their blood. This does not mean they are cured, because remember, the virus integrates into the host cell DNA. And so the virus can be between the medication and the immune system, it can stay dormant. But one of the types of cells it infects are what we call a memory cell. So our immune system has these cells that have a memory and remember past infections. these cells can live for decades. So it's important to continue taking medications, even if your viral load gets undetectable, not taking drug vacations. Because if you take drug vacations and skip medications for, because side effects are so nasty, resistance can show up very, very quickly. When we talk about HIV, we don't use the word cure. There's not a reasonable cure. Why am I saying reasonable? Because there are a small number of patients who appear to have been cured. And when I say small number, it's somewhere between 5 and between 1 and maybe up to 5. We don't know for sure. So what happened? In the early 2000s, someone called the Berlin patient in Berlin, Germany, had HIV and had a type of leukemia, a blood cancer. So treatment for blood cancer is a bone marrow transplant. Bone marrow transplants are nasty treatments. When they work, which they do a lot, it's great, but it's very, very, very hard on the body. So what happens during a bone marrow transplant is the doctors kill off the patient's bone marrow, basically destroys the patient's immune system. And then replace it with bone marrow from a donor. This donor had that rare mutation where they did not have that CCR5 receptor. The patient, after getting this bone marrow transplant, went off treatment. He lived for 13 years and eventually died from the leukemia coming back. After 13 years, there was no detectable virus, which if he was off treatment, if he'd had the virus still in his body, it should have been detectable. Now, other patients who've had this same treatment, bone marrow transplants from someone who was missing the CCR5 receptor, have gone off treatment, and while it was a slower process, eventually had rebound, which means that the virus came back. So just not having CCR5 is not a guarantee you cannot be infected. CCR5 absence is a rare condition. Finding a bone marrow transplant that matches your DNA that will be successful is a hard thing to do. So this whole process is not something that we can use to treat people who have HIV. It's physically hard on the patient who's receiving the bone marrow. It may not work. And matching bone marrow can be really, really hard. Your bone marrow has to be genetically similar enough to the rest of your body so that the bone marrow, oh, I didn't even talk about why. Why is bone marrow important? Bone marrow is the source of immune system, most immune system cells, and your red blood cells. Okay. So it's nearly impossible to do this successfully on purpose. It's been luck that it's happened in a few cases. Most people who get a bone marrow transplant stay on the HIV treatment because there is a significant risk of eventually rebounding. The best thing to do, prevention. Using condoms, using, if you know you're going to be at high risk, pre-exposure prophylaxis, although that can cause a lot of side effects, including problems with digestion, weight loss, headache. We can do post-exposure prophylaxis, which is a high dose of HIV drugs that may be able to stop the disease from becoming established. It actually works pretty well. But again, you've got to deal with those side effects, and the side effects are nasty. All right, so that's the process of infection of HIV. I think we're going to stop there. So, any questions at this point? You guys are a quiet class. All right, in that case, I will see everybody Thursday.
Thank you.
Thank you.
Thank you. Bye. Thank you.
Bye.
Thanks.
Okay, bye. Thank you.